I now call to order the Society's 2,458th meeting in the 151st year since its founding on March 13th, 1871. Good evening, everyone. Good evening. My name is Larry Milstein. I'm the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends. Tonight's lecture was originally to be given by Rahul Saini, CEO of Teliatry. Unfortunately, we learned only yesterday that Rahul has taken ill, is not able to travel, and will not be able to give tonight's lecture. Fortunately, his colleague, John Randall, has generously agreed to speak in Rahul's place. John is the executive vice president of Teliatry and CEO of Zyvex Labs, where he and Rahul have worked together closely on the technologies discussed in tonight's lecture. More about John later. I am delighted that tonight's meeting is being held in person at the Cosmos Club, including a pre-lecture reception and dinner with our speaker. We plan to meet in person regularly going forward and to use Zoom and other social media when it's advantageous in the future as well, and when it works. Please check the website often for up-to-date information. All in-person meetings will adhere to the COVID-related rules of the District of Columbia and the Cosmos Club. Proof of vaccination, including a picture ID, will be required for in-person attendance. Masking is now optional. We also suggest getting tested for COVID infection within 48 hours of the event. These requirements are subject to change with changes in the COVID-related rules of the District of Columbia and the Cosmos Club, so please check the PSW website often for up-to-date information. We will be live streaming all meetings to YouTube for our remote audience to enjoy and to ask questions in the YouTube chat box. And final versions of the recordings will be posted to the PSW website and to the PSW YouTube and Vimeo channels, where they will be permanently available without charge for everyone to view. In addition, tonight we are hosting the lecture in a Zoom webinar to provide another avenue for members and guests to participate in tonight's event. Zoom participants will be able to ask questions directly and use the Zoom Q&A to text their queries. More on that later as well. And this, of course, assumes that all the technology will work as we hope it will. The Society is grateful to the sponsors of the 2021-2022 lecture series for their support, the Policy Studies Organization in cooperation with the American Public University, and a generous donor who has asked to remain anonymous. We also want to thank the sponsor of tonight's lecture, PSW member Joe Schulman. This is a very flattering picture of Joe. So let us thank our sponsors. I am pleased to announce that the following new members have been elected to the Society. Michael Nyack, a program manager at DARPA, interested in Earth and planetary sciences and astrophysics and space exploration, who learned of PSW from PSW member David Lewis. Rahul Saini, who learned of PSW from our invitation to him to speak tonight, whose interests will be clear in part from tonight's lecture, even though the lecture itself is being delivered by John Randall. And John Randall, although in truth John is already a member, and he's known about PSW for a long time, at least since his own talk here in 2013, very hard to believe, it was that long ago. And some of his interests were described in his talk back then, and additional will be clear from tonight's talk. And we welcome them all to membership. If you're not a member and would like to join PSW or support the Society, you can do so through the PSW website using the blue Join button on the upper right-hand corner of the home page. We welcome new members and appreciate donations. Cameo Lance, Special Projects Director, standing in for Recording Secretary James Hewlin, who could not prepare the minutes or be here due to travel, will now read the minutes of the 2,457th meeting and the lecture by Elizabeth Strychowski on living measurement systems and minimal cells. Cameo.
Black mic live now. Excellent. Hello, all. On April 22nd, 2022, from the Powell Auditorium of the Cosmos Club in Washington, D.C., and by Zoom webinar broadcast on the PSW Science YouTube channel, President Larry Milstein called together the 2,457th meeting of the Society to order at 8:02 p.m. He welcomed new members, and the recording secretary read the minutes of the previous meeting. President Milstein then introduced the speaker for the evening, Elizabeth Strychowski, group leader of the Cellular Engineering Group at the National Institute of Standards and Technology, or NIST. Her lecture was titled Living Measurement Systems and Minimal Cells, Engineering Cellular Life for Understanding and Applications. Strychowski began by introducing 3 areas that her cellular engineering group is working on: advanced living measurement systems, or sensors, computation, and chassis. She explained that the Living Measurement Systems Foundry is part of NIST's larger push to modernize and automate biological labs at NIST. Strychowski highlighted that her lab is actively sharing their experience and advice on automating synthetic biology protocols by publishing a technical manuscript detailing protocols, sharing information methods on GitHub, and they are a member of the Global BioFoundry Alliance. The speaker then moved on to the topic of minimal cellular life, beginning with her goal, which is to be able to engineer a platform using minimal cells for developing and testing living measurement systems. The development of minimal cell at the heart of the lecture was led by John Glass at the J. Craig Venture Institute, or JCVI. JCVI develops minimal cells with the top-down approach by removing parts from an already functioning cell until it just barely meets the definition of life. Three key capabilities are leveraged to minimize cellular genomes: genome synthesis, genome transplantation, and a design, build, test workflow. These capabilities were applied to the organism Myocyplasma myocytes. Strychowski explained that the origin of the JCVI SYN 1.0 as being the myoplasm's myocytes genome transplanted into a related species, Capricolum. Strychowski explained that the minimal cell was developed using 8 genome segments. 8 was chosen, she admits, mostly arbitrarily. She then compared the growth of JCVI SYN 1.0 and JCVI SYN 3.0. JCVI SYN 3.0 is a subset of the genome of 1.0 and represents the first realization of the concept of minimal cells as it was originally proposed. Practical problems arise with trying to study and engineer cells that don't have reproducibility characteristics of a single cell. To address the difficulties of measuring with an electron micrograph, which requires fixing cells, which kills them, they developed microfluidic chemostats for measuring the dynamics of single cell growth and investigating what genes are required for normal morphology. They restricted the search from 428 genes that were removed from SYN 1.0 to make SYN 3.0 to only have 76 genes missing from the minimized segment 6. By focusing on segment 6, they developed a strain called JCVI SYN 3A. It has 493 genes and 19 genes not in the minimal cell version of segment 6, and it shows nearly normal cell division. She emphasized SYN 3A because it is most widely used strain among the dozens of labs now doing research with this minimal cell. The team refined the search to finding which of the 19 genes was most essential to controlling cell division. They hypothesized that cell division depended on a gene cluster that is part of a very highly conserved division and cell wall cluster. They found 7 genes known for cell division and 4 genes that encode what they think are membrane-associated proteins. The speaker then considered, as a physical object, the question of what mechanical forces are acting on the membrane that could drive constriction and scission during cell division. The lab is looking into which parameters these genes might depend on in this genomically minimal context. This will hopefully give information on what these gene products are doing and help realize this vision for understanding what every gene in the cell is doing. The speaker then reviewed recent publications and preprints involving JCVI SYN 3A and spoke about what is next with minimal cells. She advocates for an international community called Build-A-Cell. Next, the question and answer period began. One of the members asked how the speaker's work was different than that of Eric Winfrey's, or if it builds upon his work. The speaker responded that this work was part of the inspiration, that his work, pardon me, was part of the inspiration for their RNA circuits, and noted that the difference is, in general, for DNA computations, you typically make different molecular components of the computation in separate tubes and then have to mix them together. And they react that once it's done. Whereas the speaker's lab recruits the machinery of life to make the parts together in situ. A question from a member participating through YouTube asked if there was any work being done to improve the ethanol production from switchgrass metabolizing. The speaker commented more generally on the cell-based biomanufacturing, which is a NIST-supported area. She responded that there may be some opportunity for using minimal cells that have been engineered to function as living measurement systems and perhaps co-culture them or use them in some other way as part of a monitoring system in these biomanufacturing processes so that they can be competitive against petroleum processes. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. President Milstein adjourned the meeting at 9:57 p.m. The temperature in Washington, D.C. was 20 degrees Celsius. The weather was partly cloudy. And the number of streaming online viewers was 38. The number of views on the PSW Science YouTube and Vimeo channels in the first 2 weeks of the lecture was 157. Respectfully submitted, Camille Lance, Director of Special Projects.
Thank you, Cameo, so much for that excellent recording of the last meeting. I particularly like the part about the minutes. I mean, the question and answer period, which sometimes doesn't make it into the minutes. So, are there any comments on the minutes besides mine? Not having any comments or suggestions, online members can submit comments to the corresponding secretary, Robin Taylor, at corresponding@pswscience.org. And for now, is there a motion by a member to accept the minutes as read? Is there a second from a member? All members in favor? All members opposed? The minutes are accepted as read and will be posted to the website in due course, subject to any corrections that need to be made on commentary that comes to us from social media in the next few days. We now turn to tonight's lecture. And our speaker, John Randall, standing in for Rahul Saini on ultraminiature medical implants. John is CEO of Zyvex Labs, Executive Vice President of Teliatry, Executive Vice President of Nanoretina, and Adjunct Professor at UT Dallas. He and Rahul have worked closely on the developments that he will be discussing in tonight's presentation. Previously, John was with Texas Instruments, where he worked in high-resolution processing for integrated circuits, MEMS, and quantum effect devices. While at TI, John and his team fabricated the first quantum dot diode, developed the first quantum well bipolar transistor, made the first working room temperature quantum integrated circuit, and created the first lateral resonant tunneling diode. Prior to TI, John worked at MIT's Lincoln Laboratory on ion beam and X-ray lithography. In addition to his science and engineering work, John has been instrumental in developing and spinning out a number of successful nanotechnology companies, including Zyvex Technologies and Zyvex Instruments. He is the author of many peer-reviewed scientific publications and an inventor named in numerous U.S. patents. Among his honors and awards, John was elected a University of Houston Distinguished Engineering Alumnus, served on the External Engineering Advisory Board of the university, and was designated a distinguished member of the technical staff at Texas Instruments. He also is a senior member of the IEEE and a fellow of the Society of the AVS. John earned a BS, MS, and PhD in electrical engineering from the University of Houston. All questions will be fielded after the lecture during the question and answer session. John, the stage is finally yours.
Thank you very much, Larry. let me say how much I very much enjoyed discussions for dinner this evening. It reminds me what a wonderful crowd that and the members of the Philosophical Society of Washington are. And I will come back as often as I can because I really, really enjoyed conversation that we've had so far. Let me also mention a little bit about Rahul, and I think I'm about to be able to go here. Yeah. Rahul, I hired about a little over 20 years ago, and it cost me a bottle of Lagavulin scotch to do so. He interviewed, we made him an offer, he rejected the offer, and I went to our HR person, and I said, look, I don't want to lose this guy. You figure out a way to get him to accept our offer. If you do it, I'll buy you any bottle of scotch that you want. And she wanted some really old Lagavulin scotch. It cost me over $100, the best $100 I've ever spent. Rahul is a fabulous, world-class engineer, as you'll discover as we go through this discussion. I'm going to start off this way. We're going to talk about a retinal implant. And so just as a little, this is optional, this blank black slide is intended to simulate blindness. So if you will, optionally, close your eyes for a moment. Understand that humans are incredibly visual beings, and deprived of sight, we're incredibly isolated compared to our usual condition. So anything that can be done, for a long time it's been a dream of restoring sight to the blind. I'm going to explain in a rudimentary way some very great successes in doing that. Please open your eyes again. Most of you know very well how an eye works, but I want to cover this very quickly. We have a set of optics that projects the image of something that's lit that we're looking at on the back of the retina. We have rods and cones to receive that information. This information that comes in the rods and cones are turned into electrical pulses. They're carried by the optic nerve. And really, the visual realization is done back in the visual cortex of the brain. And if everything works wonderfully, then we can see in wonderful ways. It's great fun. Unfortunately, there are diseases that will kill our rods and cones, one of which is retinitis pigmentosa. And, well, first of all, and this one gives me a pointer. Yeah. Well, look at this. What a cool pointer. We've got various levels of the retina here. The rods and cones are shown here. And then in front of that, there's the bipolar cells and the ganglion cells, which carry the electrical signals, the nerve pulses up to the front of the retina, where they make a large boundary of nerves which come back through and make the optic nerve. Now, I don't want to get into a religious discussion here, but the mammalian retina is not a great example of intelligent design. An engineer would clearly have the engineer would clearly take the nerves backwards instead of pushing them forwards, and therefore we would avoid this blind spot that you all have. We rarely recognize it, but it's there where all these nerves come together and move back into the optic nerve. But it turns out to be a particular problem for some approaches to making retinal implants, and I'll get to that here in a second. The retinosa pigmentosa kills the rods and cones we can't see. Now, there's a couple of different ways, actually several different ways that we can go about fixing this problem. One way is an epiretinal bionic eye concept, where we place the electrodes that we're going to generate these electrical signals on the surface of the retina. This is a problem because on the surface of the retina, you've got nerves coming from all over the retina, and it's really hard to get decent spatial resolution here, even if you had a very fine pitch of electrodes. So there is an example of artificial retina made this way we'll cover in a moment. Then there's a subretinal approach, which gets around this problem, but it gets around the problem in sort of a draconian way. They actually go in and peel back the retina to the level of the rods and cones, try to insert some electrodes, and this gets rid of the problem of putting electrodes at the surface, but it's a difficult surgery. You peel the retina back, you do a fair amount of damage. There have been some attempts along these lines, but nothing commercialized. Now, the epiretinal approach is the approach that the one other commercially available artificial retina has taken. The Argus second sight involves a fairly complicated device that gets implanted in the eye. You actually have this wireless receiver that's implanted right next to the eyeball. The user wears a separate camera. It has to go down through a video processing unit, and then information gets fed back through a wireless transmitter into this wireless receiver here. And then this lead of cables actually penetrates the sclera, the eyeball, goes back and presses 60 electrodes against the backside of the retina. But again, because of the large number of nerves that you're exciting at once with this, it's very difficult to get good imaging. You don't get anywhere near the 60 pixels worth of information that potentially are there. What's worse is this is an extremely invasive surgery, especially for elderly people that have retinitis pigmentosa or AMD. It's a 4 to 8 hour operation. Now, a couple of hundred people have been implanted with this worldwide. These were all totally blind people before. And they are pleased that they can sense some light. But this really hasn't changed their life much. You put them in a dark room and there's a lit doorway, they can tell what direction the lit doorway is, but that's about it. So the limitations of this device actually were reasonably well known when I've got to tell a minor story here. Yossi Gross and Effie Cohen were two guys that had started up a biomedical incubator in Tel Aviv, Israel. We had a mutual friend that I've mentioned to a few of you already, Shishel Devlin Manning, who introduced us to Yossi and Effie. They came to Zyvex and saw some of the things that we were doing, and we admired some of the things they were doing. So Yossi, Jim, and Effie went out to lunch. And Jim Von Air, the founder of Zyvex Labs, doesn't like to do anything unless it's spectacularly hard and will be really spectacular when it succeeds. And he talked him into going after an artificial retina. And they were well aware of the limitations of this particular approach. And so I'm going to describe here some of the things that, well, first of all, what Rahul, to sort of short circuit the story I'll come back to, this is the artificial retina that Rahul developed. I'll say much more about that. It's a 1,000 times reduction in volume of what the company. I can. Shall I turn this off? Off. I'll get unwired here for a second. Is this better now? Okay, excellent. Good. The volume of this artificial retina, shown here between a couple of fingers, is again about 1,000x reduction. Has a lot of advantages. We'll get to that. The nanoretina concept is to get around this problem of putting electrodes on the surface by putting the light sensors on the surface of the retina, but including penetrating electrodes that reach beyond this big, thick range of nerve fibers into the either bipolar or ganglion region, and do just the very tip of these electrodes, do the electrical activation that way. And it was believed that that would give better spatial resolution. And so the problem was, with this idea in mind, plus another major problem they solved is how to get, and we'll get to this in a second, how to get power into this to avoid having to put a large battery in place. They decided to shine a laser in through the eyeballs, put in infrared photovoltaics and power the system up that way, and we'll get to that in a moment. But the platform technology since 1958, when the first pacemaker was installed, was implanted, are these titanium cans that hold batteries that are this titanium housing. They're manually laser welded together. They have low density feed throughs, a header for leads attached to them, and a relatively large size, as I pointed out. So this is one such IPG, or implantable pulse generator, and you can see the size of that comparison to a human hand. There is the size of what Rahul ended up producing. Now, at this point in time, there was not the technology, the penetrating electrodes were identified, but the technology to create that didn't exist. The technology to miniaturize things that small, you can't build things by hand and make something as small as this. It's 4 by 3 by 1.5 millimeters, about the volume of a fat grain of rice. You had to check hermeticity, which nobody knew how to do on something that small. You had to have the biocompatibility, biostability, the power and communication. He had to start over from scratch in terms of if this was going to be possible. We turned it over to the right engineer. So the concept that produces this extremely small IPG uses non-conventional biocompatible materials. We realized a 600 electrode IPG. It has the highest density feed-throughs of any biomedical device by a large margin. It uses these high-density penetrating electrodes that Rahul had to invent. And by the way, he looked at about 6 different ways to make these penetrating electrodes. There's people that work serially, and there's people that work in parallel. Rahul has a gift of working tough engineering problems in parallel. Came up with a very manufacturable process. And very importantly, went to wafer-level manufacturing, which allows for scalability. If you're building things one at a time by hand, and you need to scale up by a factor of 10, you have to hire 10x of the people. With this approach, wafer scale, you just run more wafers. It's a much, much more scalable, much more affordable manufacturing approach. And as I'll point out, a faster way to prototype. Now, the components of the complete Nanoretina or the NR600 system are this implant, and you can see here, these are the gallium arsenide photovoltaics on either side of a custom CMOS chip. Now, the only thing that this does is it turns the intensity of light on any one of those 600 pixels into modulating the current pulse frequency on one and only one penetrating electrode. So every one of the surface CMOS imagers tied into one electrode, and all it does is replaces the operation basically of the rods, because there's no color involved in this whatsoever. It's all just intensity. And so that's basically what it does, all packed into this tiny, this is a marvel, tiny package, a marvel of engineering. The other portion, of course, is a set of glasses that then send an infrared, fairly weak, but powerful enough to deliver enough power to run this. And you can not only power the device up, you can also modulate that current, that voltage, excuse me, the light intensity, and actually program the ASIC that's back here and change a little bit of the way that the intensity to, the gain of the intensity to the current frequency. Now, there was animal surgery, started off with pig eye surgery, and you can see the size of the implant here compared with the pig's eye. At first, they weren't sure how they were thinking about maybe coming in from the side of the eyeball, but after dealing with some eye surgeons, realized that basically all they had to do is the equivalent of cataract surgery. The same thing they do to remove the cornea, and so that can be done in about 15 minutes, outpatient basis, local anesthetic. It's almost exactly the same operation, just another few minutes to place the retinal implant. And it's a small retinal implant, and it's designed to go just in the fovea, the high-resolution portion of the retina. And it's really aimed at people with advanced macular degeneration, where they're only losing their rods and cones in that high-resolution portion. But it's already being tested in human clinical trials against people that are completely blind, and we're still getting reasonable results, as I'll share with you in a few minutes. Now, this was the first animal studies were really exciting to get some actual histology of where these electrodes, you can see this. Well, first of all, this is a stained slice of a retina taken from one of the pig subjects. That is this blue layer stained are the rods and cones. That's the bipolar region. This is the ganglia region, and then the thicker white stuff is where's the big mass of nerve cells at the surface of the retina. And you can see we've put almost all of these, just the very ends of the tips are the only things that are conducting. We've put almost all of these tips exactly where we want them, beneath the nerves, fiber cells, and in front of the rods and cones. And so we believe that can effectively excite the optic nerve in that way. By the way, if I didn't mention, it's a curious thing. AMD and retinitis pigmentosa kills off the rods and cones, but leaves the rest of the optic nerve pretty active. And so this effect was believed to going to be successful. And now I'm going to now show a video that was made by Nanoretina. And it actually, I'm very excited to see this video. I only saw it yesterday. I've been talking on a regular basis. I talk with the CEO of Nanoretina, Ronnie Mankiewicz, and we've been discussing this one patient. I knew him as patient 106. They protect the names of their implanted patients, but in this one we actually get to hear his name and what you're going to see. I've heard a lot about before, but it's exciting to see. But first, there's a description of how the Nanoretina NR600 works, and so I'll let this play.
Imagine going from a total blackout to being able to see lights and shapes. Introducing NR600, the system that brings hope to those who've lost their sight due to degenerative disease. Nanoretina developed a miniature implant that mimics the way healthy eyes see. Natural images pass through the glasses, enter the eye, and are captured by a miniature implant located on the retina. The image, processed by a small unit located inside the implant, is transformed into instructions. These instructions are received by an array of electrodes that are located on the retina. Corresponding signals of small pulses of electricity are sent to the retina cells. These pulses stimulate the retina's healthy cells and are transmitted down the optic nerve, conveying visual information to the brain, which perceives patterns of light. The implant is powered by an infrared beam transmitted from an external pair of eyeglasses. Gustav, 72, has been blind for half his life due to an incurable condition called retinitis pigmentosa. Since August 2020, he's been seeing again after having Nanoretina implanted in his left eye. It's a new type of vision for Gustav. Patients initially see dots of light that they detect, which form an image in their brain. With training and visual rehabilitation, these images become visual perception that continues to improve. Gustav was essentially living in the dark. Now he sees again and is gradually regaining his independence. When you think of vision, you think that it is done in the eyes, but part of it is done in the brain. It's a new language for the blind to learn. Behind eyes that don't see is a brain that does see, and a mind that thinks, and a mind that dreams, and a heart full of courage that wants to succeed and live just like everybody else.
This is really a life-changing, it's crude vision, but something that we're very excited about where this can go. And so this is something that he is obviously very happy about, and is becoming a sort of spokesperson for Nanoretina. So what Rahul has done is he's found a new way to approach making active biomedical devices. And so we've gone to a much, much smaller size. But more importantly, after he did this, and our job in terms of developing this retinal implant was done several years ago, turned over to the Israelis to work with and do the first animal trials and human trials now. But Rahul thought that, geez, there's got to be a different way. There's got to be an opportunity after making the most sophisticated interface with the human nervous system ever attempted. There's something else you ought to be able to do. And if you look at chronic diseases, heart disease, cancer, chronic lung disease, stroke, Alzheimer's, and there's a list of others, this is American, the U.S. data, 6 out of 10 Americans have at least one of these chronic diseases at some point in time in their life. 4 out of 10 have 2 or more. 7 out of 10 deaths in the U.S. are attributable to chronic diseases. And 90% of the health care cost that we spend in the U.S. go to managing these chronic diseases because they're chronic, and you have to keep supplying pharmaceuticals or other therapies to deal with this. So Rahul thought, geez, there ought to be other things we can do. And as it turns out already, even before he started thinking about this, well, heart disease already is something that significantly pacemakers have dealt with a lot of heart disease and have a very positive impact here. It still is the #1 cause of death. But chronic pain is a different thing that is not being treated very well by implants at this point in time. As I'll mention, it is, but it's a therapy of last resort, and I'll explain that in a minute. But it's the #1 cause of disabilities worldwide. 1 in 5 Americans has chronic pain. 80% of Americans will, at some point in time in their life, go through a period of experiencing chronic pain. And 75% of cancer patients live with chronic pain. There are other chronic neurological disorders that, again, some of these can be treated by implants. In particular, this list that Rahul came up with, not all of them, some of these already have active implant therapies that are significantly improving things. Some of them are, all of these are at least in human clinical trials to check efficacy of implants to deal with these various conditions. Now, we're going to take chronic pain especially and try to deal with that. Depending upon the level of chronic pain, the minor kind down at step 1, you can deal with non-opioid analgesics or NSAIDs. If the pain is a little more difficult, you need something a little stronger, you can go to weak opioids. If it's even more significant, you go to the strong opioids and methadone. And if you have really severe pain, you have to go to something much more significant like a nerve block, epidurals. And you'll note that spinal stimulators are listed here, but it is only a therapy of last resort. So very few people get benefit of chronic pain treatment through implants. And the reason is there's some significant challenges with active implants. The technology is still large, tens of cubic centimeters. You have problems doing MRIs after these things are implanted, and if you have some other injury and you want to get an MRI and your titanium can is nearby there, MRI is out. They're very costly, up to $100,000 and at the very least several tens of thousands of dollars, and that's just for the implant. The surgery can run much higher. Add-on cost. A lot of the leads can break from the titanium can. There's a lot of problems here. There's also the whole deal of the surgery. You've got to create a pocket for these implants to go in. They can get infected. There's sometimes some significant pain in that implant area later. There's regulatory, and this is something that you can't entirely get around, and for good reason, the FDA requires to make sure these are safe and efficacious therapies, but it takes 3/4 of the money to bring something to market, similar in pharmaceuticals, and something like 5+ years to market. Nanoretina we've been working on for over 10 years, and we're still not in the market yet. Finally, the whole, especially in this country, the insurance setting the cost and reimbursement cost leads to questionable cost effectiveness. The repeat surgeries add to that. There's, in other words, a number of serious limitations here. One of the reasons why we haven't improved much is because the technology has not improved very much. We're still using the same titanium cans. They've gotten a little bit smaller in size, but most of them these days are still on the order of 10 to 20 cubic centimeters. Now I'm now going to make a completely unfair comparison, but I'm going to go ahead and do that anyway. Over a similar, not exactly the same, but a similar time scale, in 1970, we went from 1,000 transistors on a chip. In 2016, we got to 20 billion transistors on a chip. Okay, now, the semiconductor industry doesn't have to put up with FDA regulations, and there's been an extremely strong drive. God bless Gordon Moore for creating a self-fulfilling prophecy that's made all this happen. And it's now a $1 trillion industry, which drives a lot. But still, the amount of progress being made in the biomedical device side has been lacking from our point of view. So that's the platform design that we've got for doing active implants currently. And the size of these things are made all the more significant when you actually see them in x-rays. Okay, for spinal cord stimulation, you can see the IPG down here. There's some leads that go up, and this is for blocking some sort of pain up on the spinal cord. This is wider than your lumbars. For deep brain stimulation, you've got to figure out a way to fit that in into the human head. And again, here's another. I forget where this goes off to, but these are sizable things. They're expensive. They're hard to put in place. Rahul conceived of a new approach, a new way to make IPGs, and it involves wafer scale technology, but and it's a very much of a platform approach. You've got a hermetic feed-through wafer where you can go from the inside of the IPG, where all the electronics and everything else are, to the outside in a very hermetic way. You can embed electronics. This works with silicon, with glass, and sapphire wafers. So there's a wide variety of things you can do here. And this has become a very synergistic, holistic approach that Rahul has taken to making these sort of implants, using exactly the same manufacturing processes, but just changing the design. You can change the electronics that you're going to use. You can these are wireless by and large, using RF, so you can put a coil to pick up the near B field. You can decide how many feed-throughs, how many electrodes you want to have, because it's typically in a transparent package. You can, if you want, use sensors that look through the glass. You can put LEDs inside for optogenetics. There's a wide range of things that can be built here with exactly the same manufacturing technology. There are other people that are beginning to realize that smaller implants are good, but so far all of them are one-off designs. This is taking a very much of a platform approach. And now here's where I definitely want to tell a story. This was in a period of time after Rahul had made the implant, the retinal implant, and his job there was done, and we were looking for somebody else that wanted some kind of implant to be used. We ran into Rob Riniker, a professor at UT Dallas and director of the Texas Biomedical Device Center. Now, Rob is also an ex-Marine. Well, you know, don't ever call an ex-Marine an ex-Marine. They're a Marine for life, semper fidelis. And so he was really interested in something called targeted plasticity therapy, which involves stimulated the vagus nerve that in a way that I'll explain in a minute, that is able to significantly affect a wide variety of maladies. And there was a vagus nerve stimulation device on the market at the time when we went to meet with him. It's now a company called Lena Nova, and they use it for treating drug-resistant epilepsy. So exactly why it works is a little bit vague, but you can zap the vagus nerve with some protocol, and for people that have drug-resistant grand mal seizures, you can get dramatic reduction. So he tried to get those. They were about $20,000 a piece, made one at a time, and he couldn't find any. He found one company that started to make them and then fell out on him. And when we came to him and said, look, do you want any sort of implants? He goes, yeah, you know, I want a vagus nerve stimulator. And we said, oh, okay, great. How many electrodes do you want? You know, because, you know, he goes, I want 2. They said, no, you don't. We said, you don't understand. We can make hundreds of electrodes. We could poke a lot of electrodes into the vagus nerve and find the ones that stimulate the way you want. How many electrodes do you want? He goes, I want 2. And so finally, we got him to explain what's going on, okay, and why this is an incredibly crude hack of the central nervous system. But the vagus nerve, again, still learning a lot about the vagus nerve. It regulates a lot of operations in the body. One of the things it does is when there's a traumatic event happens without any sort of external or internal implant stimulator, say you're a soldier and you're working in the Middle East and an improvised explosive device goes off, okay, a traumatic, huge, life-threatening thing's happening, your vagus nerve goes wild, and what it does is it sends out a huge dose of acetylcholine into the brain, a neurotransmitter. And that acetylcholine rewires the brain to some extent, okay? And so PTSD is all about people recognizing even smells that they smelled right before the explosive device goes off, and so it rewires their brain. In this case, it rewires it in a very unproductive way. But the way it was finally explained to me, it made sense to me, is it says, okay, somebody's had a spinal cord injury or a stroke, and they've had a significant loss of use of a limb. With the best physical therapy, you might, and there's a protocol for giving 10%, 20% some use of a limb. If you're down to 10% use of that limb, with the very best physical therapy, you might get back to 30% use of that limb. However, with a vagus nerve implant and a therapist with their finger on a button that can excite the vagus nerve when he wants, the therapist tells the patient, try to move the arm. So they make an effort. If the arm moves any at all, they hit the button. The vagus nerve stimulated, the brain is flooded with acetylcholine. Those portions of the brain that were effective in moving the arm now get to rewire to a larger number of nerve cells. With both the physical therapy and this targeted plasticity therapy, people have gotten back to 80, even 90% use of a limb. Okay, so and they did the control study. They punched the button randomly, or punched the button, then waited a few seconds, then told the patient to try to move their arm, or told them to move their arm, waited until a few seconds after they moved it and punched the button. No result. No, at least no change that improved the mobility of the arm. So there's a lot of other things that can be treated that way, including PTSD. So, so this is all explained to us. We finally get to it, and we said, well, how small do you want it? And he wanted it that size, a similar size that had already been used. These devices that you're seeing here in a wafer before they're split up are what is being used in ongoing human trials in spinal cord injury and PTSD, and they're enrolling some people for stroke right now. And I'm about to show you a video of one example of a quadriplegic woman, at least classified as quadriplegic, that is, I think she's a spinal cord injury patient. And again, this is not made by Teliatry. This is made by the Texas Biomedical Device Center. And this shows some of the early results in this early feasibility study. Oh, first of all, I have to say, you have to wear an external coil that feeds both power and information tells the vagus nerve when to stimulate or when to zap the vagus nerve, but it only has to be worn during therapy. You don't have to wander around with this sort of dog collar on. Only worn during therapy. So, a lot of animal studies were done. Some on rodents, more of them on dogs, and no adverse effects, no infections, very biocompatible as designed by Rahul. And so this is the video.
My name is Sarah Milburn. I am 29 years old and I am a C7 quadriplegic. I'm in grad school right now getting my Master's of Healthcare Administration. I volunteer at Baylor. I'm the medical coordinator at a dog rescue here in Dallas also. I was helping Baylor with training the PTs for this study, and then I was asking what it was for, and they told me, and so I decided to see if I qualified for this study. It was really easy. All I did was just contact the email that they gave me, and then I did a pre-consultation with the doctors that are running it, and then they said I was approved. So. It's not very invasive. Your throat might be sore for a little bit just because it's against your vocal cord, kind of. I would say a little bit of, I guess, struggle is worth a lot in the long run. So I would definitely say it was an easy recovery. I mean, it's right in your neck. So since I'm a quadriplegic, you've already had neck surgery before, but it was a really easy process. It was an in and out surgery that day. I didn't have to stay overnight or anything. The part of the study that was really cool is that it was adapted to each person. Oh, I did all the games, yeah. There was like Fruit Ninja, there was archery. The other thing that was cool is that they built like a key thing so I could work on like turning a key, as well as like weights up the side, because I said I wanted to be able to like go through Starbucks and grab a drink without spilling it. I can open the doorknobs at my house now, which I never was able to do before. I'm able to pick up things easier, but I've been able to lift heavier things, so that's good. I think it was a good experience, and I have told a few of my friends are actually the next participants.
So it's a platform. So we've made already, Teliatry has made already these devices for the targeted plasticity therapy. But again, it's a platform technology. Okay, they're working on something with 16 independent electrodes for spinal cord stimulation to deal with chronic pain and some other things. So it's just a larger number of feed-throughs, a little bit different electronics, 16 channels of electronics, still well under about a half cubic centimeter volume, and includes batteries. Okay, so depending upon the power requirements for the particular therapy required, one charge could last weeks, maybe even months, in worst case a few days, and it's recharged in the evening, if you'd like, in about 15 minutes. This is currently under development, actually, for a company. We'll come back to that in a moment or two. But again, the glass encapsulation, the printed circuit boards that includes the antenna to receive both the power, and you can send in information and reprogram if you wish, the way the therapy is done. So again, one of the more important changes here is the way that these things are manufactured. It's a wafer scale. Depending upon the size of the implant that you want to make, you can put up to a couple of hundred, at least 50 or 60. And as you're developing it, you don't have to, since you can easily change what you're putting inside the cavity, you can easily populate this with slightly different versions of the implant that you want. And so it's quicker for prototyping. And then once you get into the manufacturing regime, then you do larger wafer lots. If you need to scale up, it's much, much easier to scale up. So one of the things that Rob wanted, the guy that was asking for the vagus nerve stimulator, again, a Marine, knew lots of people that came back with PTSD and other maladies, loss of use of limbs. He wanted to democratize this, and instead of $20,000, He wanted something that could be sold for at least $5,000 or less. Okay, in volume manufacturing, the price could be significantly less than that. Okay, now I don't know what the biomedical device companies are going to charge, but this is way, way more cost effective. Another important thing about this is, let's see if I'm going where I want to go. Yeah, this is so inexpensive. You can use the same devices that you want to try in humans in the animal studies. The way the industry has worked up till now is if you want to do some neuromodulation experiments, find a new thing that might treat something, the way it typically goes in a hospital or a research institute, they get some graduate students or postdocs to cobble together a set of electrodes. They implant it in an animal. The wires usually come out, and they work at it until they get some efficacious, hopefully, result. Once they've done that, oh, we have some efficacious results. Now they go into a 2 to 3 year period of trying to develop something that can actually be implanted in a human. These are inexpensive enough. You can do your animal studies right away. Those are efficacious. You can go right into human clinical trials. So it can not only provide more reliable, much smaller, much easier to implant devices, but you can do this you can get to market in a much shorter period of time. There's more that I think Rahul would talk about this than I'm going to remember to say. I will say that there's a lot of advantages in terms of like the feed through, just even the materials used. Precious metals are not required for the lead wires and the feed throughs. You've got this great a laser welding technique that's extremely hermetic. Just a lot of advantages. Also a very holistic approach for developing the wire electronics. He's got a fabulous design team that he's put together. The last thing that I want to do is do what Rahul asked me to promise to do, is he's gotten some great support from our federal funding agencies. Actually, DARPA has been working on 2 different programs, not as a prime contractor, but as a subcontractor, in one case to Teledyne, in another case to UC Davis. And people are coming to him because of his ability to integrate things and find ways to get, so it's not just the stimulation here, but also sensing and recording. So sensors are being built into this. So with support from DARPA and also from NIH. What's next is working not only with federal agencies, but with companies. He's already lined up some very important companies. And again, what Rahul wanted to do is not become a if I've explained this before, I apologize, but I don't think I've covered this. The way that biomedical device companies work, the way they're forced to work because of the way they're funded, because of the great expense of going through the human clinical trials to get something approved, to get released. You pick one indication, you make one device, you have to get a lot of people to invest in your company because the human clinical trials and the FDA regulations are very expensive to go through. And you have to, unless you've got a lot of money to begin with, you lose control of your company. And you put in all that effort, and if it works, people make good money, the investors make good money, but you spend all this time, and it might not work, and people can lose a lot of money, and you've wasted years of your time, and you've only attacked one particular indication. Rahul wants to have this platform technology be used for a lot of different things, so Teliatry is not going to be a biomedical device company. It's going to be a biomedical manufacturer, and so we're working with separate companies that will specify what they want. He'll work with them, provide the design, give them stuff to use in animal studies, and then take it through the long and laborious process. And by the way, there's some other advantages to doing that. But so the intent is to work. We've already got some companies to work with on spinal cord stimulation, peripheral nerve stimulation, and deep brain stimulation. There's a group in Switzerland actually that used to be involved in Zyvex, Andrea Mercanzini, who already has deep brain stimulator, but he wants to do a new type that's much easier to implant, et cetera. And to continue to push out to the market this wafer scale approach of making biomedical implants. Lastly, he wanted to thank his team at Teliatry, the people that have worked with him at Zyvex, his Nanoretina team, very especially mention of Rob Reneker's team at Texas Biomedical Device Center at UT Dallas, which really pushed us into this vagus nerve stimulator. And also, the other great thing about Rahul is not only a great engineer, but he understands supply chain issues, which have really come into the fore recently. But even before the current supply chain problems came, he made sure to work that everything that he picked to go into his devices, there was a good, solid supply chain. And so he's worked very well with vendors to set all that up. That's what I think Rahul wanted me to transmit to you today. And so I will stop here and I will answer questions. If there's interest, a few people have heard a little bit about what Zyvex Labs, the company that I primarily work for, is doing. And if you want to ask questions about that, I wouldn't mind spending a few minutes. But primarily, if you have questions, let's start with questions about the work at Teliatry. Thank you very much. Here's a question right here. Oh. Do we have online questions?
All right, so we have time for some questions, and the questions can come from, I guess, our YouTube viewers or our Zoom viewers, or from our live audience here. If you're in the live audience and you want to ask a question, please raise your hand and wait for the microphone to come to you. I will call on you by the color of the foam covering your microphone. So we have a question over here from the blue microphone.
Yes, my name is Scott Matthews, and I am a member. So it seems to me that with modern microelectronics, that shrinking down circuitry electronics is maybe not such a big challenge, but shrinking down batteries is a really, really difficult challenge. And so what I'm wondering about is the possibility, one, of some form of energy harvesting, vibrational energy harvesting, light energy harvesting. But what I'm also interested in is this idea of sort of RF or wireless. You know, a lot of people now charge their cell phones wirelessly. Can any of those technologies be brought to bear on the power aspects of the things that you're doing?
Yeah, great question. Already, the wireless charging of solid-state batteries is part of one of the images I showed you, and so that's there. We've talked a little bit about energy harvesting. There are some possibilities there. It depends on the therapy, whether or not that's going to be terribly practical. Some therapies require light and intermittent stimulation, and those would be better candidates. I don't think, Rahul, do you have anything on the drawing board for energy harvesting to charge batteries? Stimulation, and those would be better candidates. I don't think, Rahul, do you have anything on the drawing board for energy harvesting? So the answer is nothing at present. At this point in time, they're largely driven by what customers are asking, but that is a possibility. And there's some interesting energy storage or energy harvesting approaches. The solid-state batteries actually can be pretty small and can be recharged quickly and have a high energy density. So that's primarily the direction I think that Teliatry is going to at this point in time. But you're right. There's some other things that control the size, one of which is if you're transmitting, especially on some of these devices where you don't have a battery and you need transmission of power, then the size of that coil that accepts the B field, it sets a limit on how much power you can deliver in that way. So there's a trade-off of things, but they've, again, got an excellent design team that can work with a wide variety of...
A question from Dorema. You have the microphone? Great microphone.
And I'm a member. And thank you very much for a fascinating presentation. First of all, I wish you all success for the benefit of all that can benefit from all the devices that you. So I also had a question regarding energy, but I have also now a question regarding the interfaces. You talked about, the interface to the optical nerve. Then you talked about this, that you can, in a sense, provide multiple chips for stimulation. And the professor that you were talking to said only I want two.
Yeah.
And again, it's an issue of interfaces. So how do you deal with that?
No,
and also before the question regarding Zydex, I would be interested to hear your comments after you answer.
Okay.
So I want to put a motion for this question to be addressed.
Thank you very much.
Thank you.
First of all, on the interfaces, especially on the Nanoretina project, they went with a fractal tinitride to really increase the surface area, allowing for great capacitive coupling, and so a lot of attention was paid to the interface there. Currently, I can pretty comfortably say it's being driven by the customers. In a lot of these cases, like in the vagus nerve stimulator, Rob was happy with just metal leads, and I think they were Rahul. What was the metal leads on the vagus nerve stimulators? Platinum. There's also some just oxides metal leads that are used sometimes. But by and large, Teliatry is working towards making the IPG. What was the metal in a lot of cases leads on the vagus nerve surface electrodes on the IPG, but they will hook up some leads that'll go off, and customers have their own specialized. They've put a lot of time and effort into working out what the electrodes look like. There are going to be some situations, I'm sure, when we're asked to provide those direct electrodes, and there's a variety of things that can be used, and already there's quite a bit of expertise on different surfaces to use there.
Hey, Rahul, it's great that you're on the line, but what we'd like to do is have you speak through your Zoom connection and not on the phone. So when you want to speak on the Zoom connection, just sort of put your hand up or something, and we'll have you unmuted, and you can ask or make your comment. Would that be all right?
No,
when you want to speak on the Zoom connection. Okay, so we're gonna try hand up or something. Meantime, we have a question from the Zoom actually. So let's see if this is going to work. Dick Garwin is on the line, and Dick, can you unmute and ask your question, or should I read the question for you?
Results on implanted sensors of human arterial and venous blood pressure.
And Dick,
Do you have any blood
unmute
pressure
and ask the question,
sensors?
or should I read the question for you?
Let me try reading it. So I look forward to seeing results on implanted sensors of human arterial and venous blood pressure. Do you have any blood pressure sensors?
Question, or should I read the question for you?
And actually, there's a DARPA program that we have right now that's looking at arterial blood pressure, and that can be integrated into these packages. Actually, I think there's already an effort to do so. Rahul, can you verify that arterial blood pressure sensors are something you're integrating currently?
We have an externalized
that can be integrated
pressure
into
sensor
these packages.
under development.
Actually, I think there's already an effort to do so. Rahul. Echo. So Rahul says yes. This is indeed something that, and this gets into something that we had a discussion about before. There's a spectacular opportunity for closing the loop on therapy. Okay, let's say you've got a stimulation that is designed somehow or another to control blood pressure, and you want to monitor that, decide when to stimulate and when not. This is a great opportunity. We can integrate those sensors. You're going to have to work out the algorithm that you're going to use to do that, but this is a great question. Thank you for that question. Indeed, arterial blood pressure sensors are on the table.
Blue microphone. Al?
My name is Al Ehrlich. I'm a member of the society. It was not clear to me if the mechanism, the detailed biological mechanisms, were in any way for the lady whose grip, turning the doorknob and lifting things up, if that was at all understood, or is it in the general thing of like this is biofeedback? That's one thing. And the second one was, has anyone been so trained long enough ago that you know that the beneficial effect does not undergo diminution with time? Is that known?
That's a great question. I think there's plenty of evidence to suggest that this targeted plasticity therapy, in fact, I don't believe a biofeedback thing that she's just using and training herself to do better. It definitely reconfigures the synaptic connections. There's plenty of animal studies that show this. Now we're getting a growing amount of data on human subjects in these human clinical trials. But it's a good question about how long this lasts. Rahul, do you have any information about, does the reconfiguring of the synaptic connections diminish over time? Do you know of any data that talks about that?
No, so I'm on Zoom. Can you guys hear me is the question, first of all. Awesome. Okay, so very, very good question. Actually, there are multiple parts of the therapy. The implant is just one part of the therapy. A very big part of the therapy is the rehab. There are specific games that are set up. There are specific software systems and hardware systems that are set up for limb rehabilitation for the patient. And that's what is driving the synaptic regrowth. That's what's driving this targeted plasticity. So you can play these games, and while you're doing that, at some point in time, based on what you are trying to induce as therapy into the system, you start stimulating the vagus nerve, and that is what is driving this targeted plasticity to empower all of the limb functionality, the motor skills.
Sure how well everybody heard that. I'm going to paraphrase what I believe Rahul said, is that it's a complicated issue. There is very clear data that there is a reworking of the synaptic connections, but it also has to be effective. It has to be tied in with the physical therapy as well, and that's also probably playing a role. I don't think we got an answer from Rahul about how long this sort of persists, but it's not. If you just do the targeted plasticity therapy, not combining it with the physical therapy, the effects aren't nearly as good. So the physical therapy has to be there as well, and it's a combination. Sure.
Great microphone. You won't be heard. You won't be on the recording.
Yeah, hi. My name's Erica Kane. I'm a member. I want to thank you for a great talk, and it's really great to see all the progress that's being made with the medical technology, and wishing you guys the best of luck on all of it. One question that I had, I see in the talk, you're showing that these circuits have been miniaturized. You've got nice integrated circuits over there and so forth. But aside, I was sort of trying to figure out how this is different in a way from, what has happened across electronics everywhere, right? Because people have moved towards wafer manufacturing, towards miniaturizing these things. And obviously those, I think those nerve attachments for the retina, that did seem like that was something that was new.
Yeah.
But I was trying to figure out what other, I'm assuming there's other things you guys have done that's unique, and I didn't quite hear the details on that, if you could talk about
I'll do my best to paraphrase what I believe, and we'll give Rahul the opportunity to correct me if he thinks that's required. But until just a few years ago, virtually all active implants required these titanium cans, and these titanium cans and everything else about the implant were made one at a time by hand. Now, doing things wafer scale, as you point out, is really old hat in terms of the semiconductor industry, and you're absolutely correct. There's nothing new about in terms of what the semiconductor industry does in terms of wafer scale, but nobody in the biomedical device industry has done this before. This is totally new to the industry. One of the things that makes Rahul a spectacular engineer is he will never reinvent the wheel. When the technology is available that will work and you can get it off the shelf, he'll do it every time. So we're adapting a lot of technologies straight from MEMS and from the semiconductor industry. But there's some very unique things that Rahul and Teliatry have done. One of which is a laser welding technique that works with glass and silicon or glass and glass that makes ridiculously good hermetic seals. Also, the whole penetrating electrode array, that technology did not exist at all. And again, Rahul looked at half a dozen different ways of doing that and finally came up with a way that actually we're accomplishing with a Canadian MEMS firm foundry called Microline, which is now owned by Teledyne Scientific. He developed a manufacturable approach there when none existed before. Also, a lot of the RF, the technology around near field, B field RF interaction had not been used in the biomedical device, and not so much in any other industry either, where you can put an implant even 5 centimeters beneath the surface of a human being and get good energy coupling in. So there's some I think relatively new, not terribly complex, but relatively new RF transfer of power and information as well that has been developed by Teliatry. I think those are the main things. Rahul, what did I forget?
So one of the key things, but first of all, again, can you guys hear me? Okay, so one of the key things in here is, let me actually try to see if my mic is.
Just talk.
So feed-throughs. So hermetic feed-throughs is the game changer for medical devices, and hermetic sealing is an essential part, right? So anything that you are embedding inside needs a housing which will not leach or leak. Anything from outside to inside or inside to outside. And that's a very, very stringent requirement, which is not there in the semiconductor or the MEMS world. And that is the key to getting very high density feed-throughs depending on what the application is. Both these technologies are really a game changer when you start looking at shrinking the size of feed-throughs. We are at the highest density hermetic feed-throughs of any compatible bio-implantable device at this point in time.
Yeah, I completely forgot that. That's actually, that came out of, I believe you could argue, the development of the penetrating electrodes, but a way of making a hermetic feed through first in silicon and later in glass. This is a major innovation that came about to make all this possible. Sir.
Blue microphone. Please, yeah, you've got it, Blue. Please stand up, tell us your name if you're a member. You're on.
George, not a member presently. I had a question dealt with the targeted plasticity kinds of experiments and also a specific application, say, for example, multiple concussion syndrome kinds of situations where there's also some plasticity. In your experiments there with the targeted plasticity, how important was placebo type effects, meaning states of mind of the patient, either before, during, or after the procedure? How important is the state of mind, placebo effect in this procedure?
That's a great question. I don't think either Rahul or I are really well positioned to answer that question. I suspect you're right that the state of mind of the individual is extremely important, especially in like the PTSD, when you have to time the stimulation knowing what the state of the mind of the person is. If you do it at the wrong time, you probably make the PTSD worse. So I'm sure that there's an effect there, but we're not the ones doing the therapy. We're providing the implant that they use to do the therapy. It's a great question. I will attempt, let me make this offer. I will attempt to talk to Rob Reneker about that very question, and to the best of my ability, I'll get that information back to Larry, and he can share that, post that somewhere. Or if you'll give me your card or your contact information, I'll try to get that information back to you.
Thank you.
You're welcome.
I have another question at the back there.
Thompson, I am a member. It sounds like most of what you've described, most of the devices that you've described, kind of take an external stimuli and generate a pulse. And I'm wondering about the challenges or barriers to, taking a signal from within the body and transferring it somewhere else within the body. So I'm thinking about, say, bridging a spinal cord, a severed spinal cord, or something like that. Is there any challenge to that, or fundamental barrier to that?
Yeah, there has been some really interesting work on nerve regrowth, but I think it's my guess, and actually I sat next to a orthodontist that asked me about a very similar question. I don't think that we're going to be effective in terms of a complex set of nerves of being able to sense the nerve. Let's say there's a significant nerve break. I don't think we can put a set of electrodes in one side. I don't think we have enough electrodes to put a set of electrodes in one side and then transmit the information over to another set of electrodes. I'm not saying that's impossible. I'm just saying that most major, like the vagus nerve's got like 80,000 different neurons, axons that are carrying different signals. And there's different kind of nerves. There's sensory nerves, there's motor control nerves. So I'm not saying that's impossible. I just don't think that we're going to, if I understand your question right, that we're going to be able to put a set of electrodes on one set of nerves and then be able to transmit that information either wirelessly or some other way and put that same information into another set of nerves in a particularly efficacious way. I'm not saying that's impossible, but I think that would, at this point in time, that would be pretty tough. This would be my call. Rahul, any quick question, any quick opinion about that?
I do. This is a very interesting question, actually. This is why the DARPA BG Plus program exists, the Bridging the Gap Plus between the brain and the body for spinal cord injury patients. And I can't disclose specifics, but we are working within the BG Plus program for a very similar concept of nerve growth outside of the electrode systems that we have, outside of the IPG systems that we have. You will probably have to contact DARPA to disclose more information about that. We are not allowed to do that.
Lloyd, we have a question here. Blue microphone.
Mitchell, I'm a member. I think I'd restate more clearly what you were trying to imply about the difference between the titanium can and your glass technology, and that is the reason your technology and device field didn't shrink is because of regulatory issues. You have to seal this so that none of the things leak out, and you've developed that. So that's why you're able to get away from these giant titanium cans. And that's your biggest probably breakthrough, I would imagine, in the device area to start with.
Certainly, I would in general agree with that.
So I'll stick a question in here. Do you know anything about what Neuralstem is doing? The Elon Musk company that's developing brain implants?
That is a question for Rahul.
Okay, and I only ask because they have this technology for making really, really, really tiny electrodes that might be useful in your artificial retina, things like that.
Rahul, want to make a comment about Neuralink?
Yes, can you hear me? Okay, so yeah, we often get compared, especially on the Bionic eye side, with Neuralink. Neuralink device is about 20 times bigger than the Nanoretina device, so I can understand the comparisons there. But again, there is nothing in the FDA side which allow you to have a brain-machine interface without an indication at this point in time. The FDA guidelines for a brain-machine interface just came out, I think, sometime earlier this year or late last year.
Not the size of the electrodes that they use versus yours.
Larry, I did not hear you.
Oh, I think my question was aimed more at just the technology and size of their electrodes versus your electrodes, not comparing the companies.
Oh, yeah. So electrode size, so they have wires like hairs that are coming out, which are much, much longer, and they need a robot to implant those electrodes inside the brain. They are only penetrating, a few centimeters in the brain, not like deep brain stimulation leads that go deeper into the brain. So they're only on the periphery of the brain. But again, it's a good start. I don't know if that's the best solution. We have a non-invasive brain-machine interface program with DARPA. At this point in time, we're in phase 2 of that program to read and write into the brain. Of course, I'm biased towards the approach. I don't like the invasiveness of a brain-machine interface into the brain. And of course, if it's non-invasive and we can still do the read and write work from outside, that will be good. But their electrode wires are pretty small, and we still need to see data coming out of them for the reliability side, how reliable that is in the long run. The materials that are being used for a few tens of micron electrode wire, how long do they sustain and survive inside the body? So lots to look forward to, really.
Yeah, let me just throw in and say that it's probably a good idea to pay attention to what any Elon Musk company is doing if you're in that area. But I would also say that competition is a wonderful thing, and the more companies that move in the direction of miniaturizing and improving and going to different manufacturing techniques that are much more efficacious, that's a good thing for the healthcare industry. And if Teliatry is effective in generating quite a few competitors following the path that Rahul has blazed, then that's a great thing, I think.
Another question from the blue microphone.
So we have right up the road from here the Bethesda Navy Medical Center and the Walter Reed Army Medical Center, and they're doing amazing work with active prosthetics. And I'm wondering, do you see a role for these implantable devices to be able to interface, so an amputee can interface with an active prosthetic?
Absolutely. Actually, I know very well the guy that made the biggest impact in advanced prosthetics, and that's Jeff Ling, who was a DARPA program manager that we actually had a small role in the advanced prosthetic program that he launched that looked at, gathering nerve information and passing it on to an artificial prosthetic. We were just a little bit too far out for the timelines that he wanted, but Jeff Ling is one of my heroes, and he has played a strong role in affecting what Zyvex wanted to do in that area. And yeah, I do believe that there's some great opportunities for you putting electrodes, and this is something for sure for control of a limb, that we could insert electrodes, interpret that information, and pass it on to an artificial limb of some sort, or to control things that are not limbs, not a brain interface, but a human nervous interface with peripheral nerves that can still be used to control things external to the body. There's a lot of really interesting things. Let me mention one other thing about, say, an offshoot thinking in the future of what we can do with the Nanoretina technology. If we can pump information into the optic nerve that the brain interprets as imaging, and we can increase the amount of information we can feed into it, that doesn't have to have anything to do with an image in front of us. You could download HBO movies directly into the optic nerve. You could do artificial, you could do augmented reality, you could do virtual reality in a really amazing way. But that's somewhere way further down the future, because right now we've got 600 pixels worth of information, not the unbelievable number of pixels in your actual vision.
Get a microphone. Get a microphone.
Is this the guy who was a DARPA manager for quite some years?
Jeff Ling, yes.
Yeah.
Unbelievable. Well, first of all, MD, surgeon, was active and operating in the first Gulf War, seeing soldiers that had lost limbs. He's also a PhD molecular biologist coming out of Cornell, and he was, I think he was working on synthetic opioids at Cornell, and a colonel in the Army, and a DARPA program manager.
And a paratrooper.
Yeah, just an amazing guy.
He does it all.
Really. Yeah.
I think he actually gave a PSW talk, but I don't think it was videotaped.
Oh, he did talk here?
I think he did. We did have somebody talk on prosthetics who was from Walter Reed, and he was talking about, was Walter Reed or Neville? I don't remember exactly. Anyway, we can go look and see. I think there's only minutes, though. Are there any other questions? So the questions from outside universe? If there are no more questions.
We did have something on the table, and that was a brief discussion of Zyvex Labs stuff.
Wait.
Oh, I'm sorry. I'm waiting.
What? Say that again. Do we have another question? A YouTube question. Well, I think Cameo has to read that out loud. You're on.
1, 2, 3. Excellent. There is a question from the web. Ben Balweg. If you could let us know if you're a member or not, that would be great as well, Ben. The question is at hand. Geoff Ling's a big fan of the recently passed ARPA-H. Are medical devices staying with DARPA or transferring to ARPA-H?
I don't know the answer to that at all. I might be able to ask Stephanie Tompkins, the DARPA director, but let me say this. I'm a huge DARPA fan.
Any other questions? We have a hand raised here. Let's get a microphone over there. The blue mic. Could you stand up and tell us your name and if you're a member?
Yeah, my name is
Thank
Alan
you.
Gottfried. I'm a guest. Talking about the future, reaching out there in terms of what will be in terms of senses. What about stimulating or enhancing thinking, cognition, reasoning, and logic or intelligence?
There has been some thought that the targeted plasticity therapy could enhance learning. In fact, if you think about it, rewiring the brain is sort of long-term memory, and there has been some work done to suggest that with targeted plasticity therapy, you might be able to improve learning processes. After some early encouraging results, the last time I asked, particularly Rob Reneker, who knows this technology very well, is that the human brain is really pretty well optimized at learning. And so while there were some, looks like there were some advantages there, so far I don't think there's any really firm data that this really enhances learning in a really significant way. Having said that, this is definitely understudied. It's certainly playing by stimulating things and changing the amount and number of active neurotransmitters in the brain. There may be people that have some deficiencies in that you could improve and help. It's a great question. I'm not the right guy to answer it, and I don't think Rahul is particularly either. Our job is, Teliatry's job, is to make the equipment that people can do the research with and see if that, it's a great question and a great place to possibly go. And there has been some excitement about that, but my best understanding is after some initial excitement about that, it looks like something that hasn't borne out a lot of real positive results that seem to be, people are going to follow it up. And I hope you're right, that we can help learning, but early results are not great, I think.
It's a great question. I hope you won't mind the following attempt at humor, but I don't think live streaming Netflix to an artificial retina is going to help improve people's intelligence. Blue microphone, Dorena.
If I want to add to that, maybe this will
Could
be
you speak
for,
slowly and clearly so we can
maybe
pick
this
it up?
will be useful, not for learning in general, but for older people to maintain their mental acuity. Maybe that would be
There is, according to Rahul, there are some active brain stimulation therapies that are looking to reduce Alzheimer's effects. Right. And so that's one area. And it's not targeted plasticity, but there are some chances, especially for things that diminish the brain, like Alzheimer's, to perhaps make an impact there. That would be excellent and exciting.
So I want to second my own question about your comment that you said you had something interesting to tell us about Zyvex.
Okay. Yeah, briefly, I'm the CEO of Zyvex Labs. We were set up by Jim Von Eer, who was a fan of Drexler and Feynman, and back in 1997, when nano was not even a cool buzzword, he not only decided to start a nanotechnology company, but a nanotechnology company focusing on the end game of nano, where you can control where atoms and molecules go with a high degree of accuracy. And so the main technology we've been working on is hydrogen depassivation lithography done with a scanning tunneling microscope, where we can do atomic precision removal of a monolayer of hydrogen on top of a silicon 100 surface. And actually, there's somebody, a member here sitting in the audience, that has strongly affected the direction we've gone, and that's Bruce Kane, who back, when did you, 98, suggested a way of making a quantum computing qubit, which I believe his first suggestion was to use the nuclear spin in a phosphorus atom embedded in silicon, and use some gates to control both, to set the spin, the nuclear spin, and also gates to control the interaction between one qubit and another. And so it takes the way that the phosphorus atom is placed, and Michelle Simmons at University of New South Wales, I believe a speaker here previously. She is not. Oh, where did I see she was a speaker? I thought it was here. Okay. Michelle showed that you could use the hydrogen depassivation lithography in ultra high vacuum, use dose with phosphine. The phosphine will stick where the hydrogen's gone. So hydrogen's got the 102 by 1 silicon surface. For every surface silicon atom, there's an unsatisfied covalent bond. You can throw atomic hydrogen at that and then passivate each of the surface silicon atoms, come back and remove the atoms that you want, dose with something that will stick where the clean silicon is, and in this case, a phosphorus atom that will go in substitutionally for the silicon. And then, so we've been working on improving the technology of scanning tunneling microscopes, which are reputed to be, by the popular press, the first way that humans ever saw atoms, which was wrong by about 30 years. The field ion microscope in 1951 saw atoms at the end of a tungsten tip, but the popular press rarely gets things all that right. So a Nobel Prize was won by Binning and Rohr at IBM Zurich, inventing the STM. Joe Lighting and Fade Navoris developed a technique where they showed they could image the silicon surface and see where all the hydrogen atoms were, and then remove hydrogen atoms with atomic precision. The problem is, and that's been demonstrated back in the early 90s, mid-90s, moving forward. Joe Lighting has continued to do that. There's a number of practitioners. But the basic STM technology is pretty much the same. The digital electronics have improved a little bit, but they use the same PI loop. They use the same piezo tubes that have horrible creep and hysteresis. The PI loop is designed to keep a constant current between the tip and the sample, but it gets unstable and crashes the tip all the time. Automation is horrible, and so we've spent a huge amount of effort improving that. We've got creep and hysteresis correction. A brilliant control system guy, Reza Mohammadi, has figured out a way to keep the tip from crashing. We can do automatic lattice lock to the surface. We have a high degree of automation. We're working with Oak Ridge National Labs to automate using AI and machine learning to do better image recognition of the silicon surface, to do automated error correction for when we make a mistake doing the lithography. So we're putting a lot of time and effort into developing atomically precise fabrication, something that a lot of people were excited about early on the Foresight Conference. There's other people, a lot of people were working on atom manipulation, but we're the only company that I know of, other than Bob Walco up in Canada, who's doing sort of similar work, that's really working on trying to do atomic precision fabrication. And so that's primarily what Zyvex Labs does. There's a lot of other ridiculous details I could give you, but that's the short story.
Okay, any more questions?
So wait, before we go.
Wait, before we go.
Before
that was my line.
I told a quantum joke during dinner, and so I'm going to repeat that. The one that had Cammie almost fall out of her chair. So what's the difference between a quantum theory conference and a fashion show? The difference is that at the fashion show, the models actually work.
And with that, before you go, we have a small token of thanks. We have a signed copy of Volume 1 of the Philosophical Society of Washington, Volume 1 of the Bulletin of the Philosophical Society of Washington, in which you will discover who the original founders were, why they founded it, why they decided to name it the Philosophical Society, having nothing to do with Kant or Hegel or Plato or Socrates, maybe Aristotle. But, and also, before you go, there's also an interesting article on there to tell you how to calculate the value of pi to 30 decimal places.
Excellent. And I'll bet one of those is for Rahul.
It is. And thank you for Rahul for preparing the slide deck and prepping you to give the talk. And for agreeing to give the talk in the first place. We have a volume for you as well. They're both signed on behalf of the members of the society.
Well, thank you very much. Again, I have enjoyed this immensely. You are a great crowd of people to talk to and from the podium, but also I've enjoyed all the discussions I've had. I should be back for your June 3rd.
Tahan. Yeah, Charlie Tahan.
Yeah, I plan to be there, be here for that. I'm really looking forward to
June
that.
June 3rd, you are correct, number 2460.
So again, thank you very much. This a great place to come and a wonderful organization. Thank you, Larry.
Thank you,
Rob.
John.
Thank you, Candy.
And before you all go, we have a few closing remarks. We're planning for the next meeting to be in person. In fact, it's going to be in person no matter what, because it is the Joseph Henry Annual Lecture, which is our annual Black Tie Preferred event, and we hope that many will come. I should say the tuxedos are strictly for fun. Well, we're a little serious, but you know. Anyway, the speaker will be Bob Ballaram. He's a JPL Caltech Chief Engineer of Ingenuity. And of course, he'll be speaking on Ingenuity, the first flying machine on Mars. And the rest of the fall, well, it's the spring. The rest of the spring schedule is here. As we just mentioned, the 2460th meeting will be on June 3rd. The speaker will be Charles DeHaan. He's the head of quantum computing at the White House Office of Science and Technology Policy, and also chief scientist at the Laboratory of Physical Sciences. He'll be speaking about quantum computing and the National Quantum Computing Initiative. The 2461st meeting will be on June 17th, and that will close out our spring lecture series. The speaker will be Shabita Satyapal. She will be speaking on supermassive black holes. Shabita is professor of physics and astronomy at GMU. And the fall lecture series will open with number 2462 on September 9th. And the speaker will be Drew Weissman of the University of Pennsylvania, who is one of the key, well, who did one of the, how to put this? His work on mRNA modifications is one of the key developments that allowed the development of the COVID vaccines that most of us have taken. And he'll be speaking about the development of RNA vaccines. Please check the PSW website often for up-to-date information on meetings and whether lectures will be in person or via Zoom. COVID and COVID-related public health measures are constantly changing, and PSW is adjusting plans accordingly. Join us for these meetings, and also please note a new feature on the website. You can subscribe to the PSW calendar for automatic updates to the schedule. As they become available, just click on the calendar icon on the PSW homepage and follow the prompts. Finally, let's thank tonight's volunteers for producing this meeting: Robin Taylor for audiovisual, live stream, Bill Goodman for the same, Cameo Lance for the minutes, standing in for recording secretary James Hewlin, and for handling the YouTube live chat. Mark Clampin and Laurel Kane for checking in room management. Robert Thompson and Laurel Kane for doing the cameras. Rosemary Collins for handling mics, to some extent. Other friends, and myself, Larry Milstein, for presiding. And with that, I will now adjourn the 2458th meeting of the Society. I wish everyone a good morning, afternoon, evening, or night, wherever you happen to be. And I hope to see you or hear from you next time. Meeting is adjourned.